After revision, let us start studying about the proteins today. So today we shall study about proteins. If we study the various applications or various uses of the sugars, the important use of our sugar is our genetic code that may be of either DNA or that may be RNA contains two, the genetic code contains two aldopentose, which is a sugar. Either it may be the D-ribose or 2-deoxy-D-ribose. These two sugars. See, this D-ribose is present in DNA and D-ribose is present in RNA. These two are sugars in nucleic acids. So our DNA and RNA are the nucleic acids, and thus we understand a very important aspects of sugar and their applications. Now we want to study about the new part, that is what it is known as proteins. So let us study about proteins. Proteins are genetically generally the polymers of amino acids. Now, how does the word protein comes into the existence? So word protein comes into the existence by some of the means, and these means are based on word from word protein comes from the Greek word proteos. The word protein comes from the Greek word proteons, which means the primary or holding the first place. So the word protein comes from the Greek word proteos, and the meaning of the word proteos means primary or holding the first place. So my dear students, from this itself, we can easily understand or we can realize that here we have the protein, which is the very important, holding the first place in our diet. So we should be regularly eating proteins in our diet, so that, or thus proteins are the most important part of our diet, and they are holding the first position, or they should be holding the first position in our diets. Proteins are generally the polymers of amino acids and building block of our body. So all our body is mainly generated from the proteins, and proteins are the building block of our bodies, and our growth also is because of the protein. So growth is possible only due to the proteins. Proteins were come from, protein word comes from the proteose, which means important, and this way we really understand the proteins are very important for our body. Now let's move forward. Proteins are the most abundant molecules of living system. Chief source of proteins are, from where do we go get proteins, or what are those foods that we eat so that we can get protein? First is milk, another is cheese, third one is pulses, fourth one is peanuts, and fifth one is fish. Proteins are the polymers of amino acids. So to understand the protein, we need to understand amino acids. So proteins are the polymers of the amino acids, and to understand protein, we must understand amino acids. Let's study about the amino acids. Amino acids, if you look at the names, you will come to know that amino acid is made up of the two words amino and acids. So amino word comes from the group amine, NH2, and acid is the word that comes from the functional group carboxylic acid. So for acid, we can easily understand the COOH group. So amino acids are the organic compound that has both amine and carboxylic acid group present simultaneously. So in amines, my dear students, we have both the groups, either it is the amine group or carboxylic acid present simultaneously in the amino acids. So we can write the structure of amino acid in this form, that this R, CH, NH2 and COOH. This NH2 and COOH must be present in the structure of protein. This R may vary for the various amino acids. Please remember that only the structure of R varies for the various amino acids, but this carboxylic acid and amine group must be present. Now, if we start giving the priority to the functional group, the carboxylic acid gets the first priority, and that is why the number in carbon that is present in the carboxylic acid should be given 1, and this CH group get the second number. From the general or from the trivial way of giving names, the first carbon that is bonded to the functional group is known as the alpha carbon, and that is why the carbon present in CH group of amino acid is also known as the alpha carbon. So if NH2 group is present on alpha carbon, it is called the alpha amino acid. So as you can see that here, my dear students, we have amino group present on the alpha carbon, and that is why the amino acids that where the amine group is present on the alpha carbon, we call it alpha amino acid. And only alpha amino acids are obtained from the hydrolysis of proteins. So please remember that when we do the hydrolysis of proteins, all the amino acids that we get, they are all the alpha amino acids, and we can easily understand that proteins are made from the alpha amino acids only. They may contain other functional groups also, depending upon the group R. So what is R, on the basis of that we can have the other functional groups. We have many amino acids, and amino acids were not known by their IUPAC names, but they are known by their trivial or common names. So my dear students, please remember that amino acids are not well aware or not well known from their IUPAC names, but amino acids are known using their trivial names or common names. Trivial name of the amino acids represents its property or its source from where these amino acids are obtained. Please remember that the trivial name of amino acid, depending upon either from where these amino acids can be obtained, that means their source, or their property or the particular property of that amino acid. Let us understand about the amino acids. First amino acid that we want to understand is glycine. Glycine, which is named from the Greek word glykos, which means the sweet taste. So the word for amino acid glycine is given from the Greek word sweet taste, and this glycine is a sweet amino acid. Same way, my dear students, we have another amino acid known as the tyrosine, was first obtained from the cheese. And in Greek, tyros means cheese, and that is why the amino acids obtained from the cheese is known as tyrosine. Please remember. Apart from this alanine, which is an amino acid where R is equal to CH3, valine, which is CH3 twice CH bond R. So your R is equal to CH3 twice CH, which is the isopropyl group. So when in the form of R, we have the isopropyl group available, my dear students, we generally consider that kind of amino acid as a valine. So these are the way by which we know the various amino acids. These amino acids are known by their short names also. So we even understand the amino acids by their short names, short symbols. They can be even understood by their short symbols. These short symbols also are of two types. So for amino acids, we can have the various symbols, and these symbols are of two types. Number one, 3-letter symbol. So we can use the symbol for amino acid as a 3-letter symbol. We can use the amino acid as a 1-letter symbol. So using this, we can have the glycine, which is having either the symbol GLY, or we just remember it as a capital G. Same way, my dear students, if we have the amino acid alanine, that is having R is equal to CH3, so we either remember it as ALA, or we just remember it as a capital A. Same way, my dear students, if we have the amino acid valine, we, by three-letter symbol, we call it VAL, or just we call it VA. So my dear students, these amino acids can be easily understood by the single letter or three-letter names. Amino acids are classified on the basis of the number of NH2 group and the number of COOH group present in them. If amino acid is equal to the carboxylic acid, the amino acid is going to be the neutral amino acid. If amine group is more than the carboxylic acid, then the amino acid is going to be basic in nature. And if amine group is less than the carboxylic acid group, or if the number of amine group is less than the number of carboxylic acid group, the amino acid is going to be acidic in nature. Once again, my dear students, on the basis of the number of amine group or number of the carboxylic acid group present into the amino acid, we can derive or we can write the amino acids or we can determine the nature of the amino acids. The nature of the amino acid can be determined easily on the basis of the amine group and the number of the carboxylic acid group present. If the number of amine group is equal to the number of carboxylic acid group, we will have neutral amino acid. If number of amine group is more than the number of carboxylic acid group, we will have basic amino acid. If number of amine group is less than the number of carboxylic acid group, we will have acidic amino acids. Again, those amino acids which are synthesized in our body are called as non-essential amino acids. Please remember, those amino acids which are synthesized in our body are called non-essential amino acids. Amino acids, while those which are not synthesized in our body are called essential amino acids. So once again, my dear students, we can classify these amino acids as a non-essential amino acids and essential amino acids. Non-essential amino acids are the amino acids which are synthesized in our body, whereas the essential amino acids are the amino acids which are not synthesized in our body, and that is why we have to eat them regularly in our diets. So those amino acids which are not synthesized in our body are called essential amino acids, and these essential amino acids are supplied to the body from the food sources, and so we should regularly intake those essential amino acids to our body by the food intake. Amino acids are colorless. Please remember, these are the physical properties of amino acids. Amino acids are colorless, crystalline, water soluble solids with high melting point, but they behave as a salt rather than a simple amine or carboxyl group substances. So please remember that amino acids are colorless, they are crystalline solids, they are generally water soluble, they have high melting points, and instead of simply existing as an amine or carboxylic acid, they remains as a salt in our body. In aqueous solution, COOH group loses H plus and becomes COO minus. Same way NH2 group accept this H plus and become NH3 plus ion. And we get amino acids with the presence of both negative and positive charges. This makes amino acid remain present as a ZWITTER ion. In the last chapter, even my dear students, we have studied about the sulfonilic acid. This sulfonilic acid has got two groups. Either it was SO3H or it was NH2 group. This SO3H group loses its hydrogen, and this NH2 group become NH3 plus and it becomes SO3 minus, and we consider this as a ZWITTER ion. So ZWITTER ion is an ion that has both positive and negative charge available simultaneously into the compound. Same way, my dear students, in amino acids, the acid group loses its H plus and amine group gain this H plus and turns out to be NH3 plus amino acid. The group COOH turns out to be COO minus, and thus in amino acid, in aqueous solution, amino acid exists as a ZWITTER ion in which both the positive and negative part present simultaneously. So in aqueous solution, the amino acid is present as a zwitter ion in neutral, but so which is neutral in shape because the positive and negative ion both present in simultaneously. That is why it is neutral, but contain both the positive as well as the negative charges. So zwitter ion of amino acid is actually neutral in state. It is not acidic or basic, but it contains both the positive and negative ion simultaneously. In zwitter ionic form, amino acids shows the amphoteric nature. Amphoteric nature means these amino acids, they behave or they react with acid, they even react with the base, and that is why they behave like an amphoteric behavior as they react with both acids as well as they react with the base. So zwitter ion exists in both acidic as well as basic condition. You can see here, my dear students, on your screen that we have an amino acid. When it is added to water, it turns out to be zwitter ion, where you have the COO minus group as well as the NH3 plus group present into the zwitter ion. In basic medium, you can see that this OH minus combines with the hydrogen of NH3 plus, and it turns out to be RCH2COO minus. But in acidic medium, it turns out to be RCHCOOH, because in acidic medium the H plus given by acid is accepted by the COO minus, and it turns out to be COOH, and we have NH3 plus ion present into it. That is why you can easily understand that this zwitter ion can react with the base, this zwitter ion reacts with the acid. In basic medium, it produces a COO minus ion, and in acidic medium you can have that NH3 plus ions, and that is why it can react with both acid as well as base, and that is why our alpha amino acids are generally amphoteric in nature. Thus it shows amphoteric nature of the amphoteric nature that can react with both acid as well as bases, with both base as well as acid. If aqueous solution of amino acid is kept between two electrodes. Now if we want to put the aqueous solution of amino acid in between the two electrodes, that is positive electrode and negative electrode, and pH is kept between 5.5 to 6.3, zwitter ions are not attracted by the electric or magnetic field. Now this is again a very important characteristics. If we put this zwitter ion, which contains both the positive and negative ion, and if the pH is kept between 5.5 to 6.3, these zwitter ion are not attracted either by the positive electrode or by the negative electrode, or in the magnetic field by the north pole or the south pole. The pH between which, now this is the definition, please learn it well. The pH between which the zwitter ions are not attracted by the electric field or by the electrode, it is called the isoelectric point. Please learn, my dear students, the definition of the isoelectric point. What is the definition of the isoelectric point? That I am going to share it with you once again. Please remember that the definition of the isoelectric point, the definition starts from here. The pH between which zwitter ions are not attracted by the electrodes, it is called the isoelectric point. So isoelectric point is related to the pH of the solution. The pH between which these zwitter ions are not attracted by the electrode, it is called the isoelectric point. And isoelectric point for our amino acid, the isoelectric point for the amino acid is between 5.5 to 6.3 pH. So even the value for isoelectric point for amino acid is known, and that ranges from 5.5 to 6.3 pH. L and D form of the amino acids. When amino acids are written in the Fisher projection, where, see, amino acids can be written by the Fisher projections, where you have COOH and H are on the vertical positions, whereas R and NH2 we put it on the horizontal position. So if NH2 group is present on the right hand side, we consider it the capital D form of the amino acid, and if NH2 group is on the left hand side, we consider it as a L form of amino acid. This is how the capital D and capital L form of the amino acid is determined using the Fisher projections. Please learn it well, my dear students, that on the basis of the position of the amino acid group, we determine the capital L and capital D position, or capital L and capital D isomers of amino acids. Most oxidized carbon is kept upward. You can see that the most oxidized carbon that is present into the COOH group, which is kept upward, and hydrogen at downward. You can see in both the cases we have put hydrogen downward and COOH upward. But if NH2 is on the left side, it is called the L form, and if NH2 is group on the right side, we call it capital D form. This is how we understand. Except glycine, please remember, except glycines, all the naturally occurring amino acids are optically active. Except glycine, this is the important point to remember, except glycines, all the naturally occurring amino acids are optically active. Since the alpha carbon of the amino acid has 2 hydrogen in glycine, since the alpha carbon of amino acid, glycine has 2 hydrogen. So it is not optically active. You need to remember, my dear students, that for glycine, R is equal to H. So we generally have the amino acid as RCHNH2 COOH. But for glycine, my dear students, we can write it as HCNH2 COOH. And again we have H here. So both the group present on this carbon are same, and that is why it is optically inactive, or else all the other amino acids, they have this carbon, which is chiral carbon, and due to the chirality, they show the optical activity. Once again, I am repeating for you, my dear students, that except glycine, all the naturally occurring amino acids are optically active, and glycine is not optically active because there is 2 hydrogen present on the same carbon. Most naturally occurring amino acids have L configuration. Please remember that most of the amino acids occur in the nature, they have L configuration. Now we want to study about the structure of proteins. So we have seen that proteins are the polymers of amino acids. So proteins are formed by using the amino acids. You can see here one amino acid. This is amino acid number 1. This is the amino acid number 2. Both they combine by removing the water molecule. H and OH gives us H2O. And thus by the removal of an H2O molecule, we get the peptide bond. Peptide bond is the bond that is formed into the proteins, which is CONH bond, or bond formed between C double bond O group and NH group. This kind of bond which is found into the protein is known as the peptide bond. Please do not misunderstand it with the process which is known as the peptidization, which we have studied into the surface chemistry, where we have studied that during the process of peptidization, the colloids are formed, or peptidization is the process of formation of colloid by shaking them with the proper solvent. But here here this peptide bond is the bond that is formed between the CO and NH into the proteins, and it is the bond which is known as the peptide bond. It is an amide bond because it is formed between C double bond O and NH group, and that is why this peptide bond is also called the amide bonds. This way, the amide bond formed in the proteins between the alpha amino acid is called the peptide bond. This is the definition of the peptide bond. It is also known as the peptide linkage that is found into the proteins, my dear students. When a peptide bond is formed between 2 alpha amino acids or amino acids, the peptide formed is called the dipeptide. Please remember, my dear students, when a peptide bond is formed between 2 amino acids, between 2 alpha amino acids, the product formed is called dipeptide. For example, here if glycine, that is R is equal to H, and alanine, where R is equal to CH3, combines, let us see what happens. Here you have glycine, where you have CH2 is bonded with NH2 and COOH, and here you have alanine, where R is equal to CH3. So this is how both these combine with each other, the water molecule from H and OH are being removed, and there is a CONH bond formed between these two. So here from this you have C double bond O, and from this NH part come and combine, and thus this CONH bond is formed between the amino acid, and as it is made up of two amino acids, it is known as the dipeptide bond. Remember, my dear students, this is the dipeptide bond, or this is called dipeptide because two amino acids are bonded to each other into this, and this CONH linkage is known as the peptide linkage or peptide bond. Now, if the third amino acid is combined in the dipeptide, it becomes tripeptide because it has three amino acids bonded to it, and there will be two peptide bonds. So in dipeptide we have one peptide bond, in tripeptide we have two peptide bonds. If there are four amino acids combined, we will have tetrapeptide, and it will have three amide bond or three peptide bond. And the alpha amino acid in it, same way, if we have tetrapeptide, pentapeptide, hexapeptide, and so on, my dear students. And if we have number of alpha amino acid, monomer is more than 10. Please remember, if we have the number of monomers of alpha amino acid is more than 10, we will call it polypeptide. So here we need to remember that if two number of polypeptide for AA is, if the number of polymers or if the number of monomers present in peptide is, or number of monomer of amino acid present in the peptide in the protein is n, then there will be n minus 1 peptide bond present into the polypeptide or dipeptide. So, my dear students, we can easily understand that the dipeptide has one peptide bond, tripeptide has two peptide bond, tetrapeptide has three peptide bond, pentapeptide have four peptide bond, hexapeptide has five peptide bond, and then n polypeptide, which has n monomer, has n minus 1 peptide bond present into it, and thus we can easily understand that the number of peptide bond present into polypeptide is equal to n minus 1, where n is equal to the number of monomers present into the polypeptide or protein. So polypeptide is also known as a protein. Generally, if a polypeptide has 10,000 U, that is the molar mass or molecular mass. If the molecular mass of polypeptide is 10,000 U or more than 10,000 U molecular mass, the number of amino acid is more than the number of monomer is more than 10, then more than 100, my dear students, then it is called protein. Please remember, my dear students, that if a polypeptide has the mass more than or molecular mass more than 1000 U or unified mass or AMU, and if the number of monomer is more than 100, in that case such polypeptide is known as protein. But there are some exceptions. Let us study the exception in this case, my dear students. There is an exception. What is the exception? Insulin with a even number of amino acid, that is 51 amino acid only present into the insulin. So in insulin, which is having only 51 amino acid present, but it is called proteins because it has a well-defined conformation of proteins. So this is the exception, which is insulin. Insulin is an exception in that case that it has only 51 amino acids present as a monomer in it, but it has well-defined structure of conformation of protein, and that is why insulin is considered as a protein, though it has got only 51 amino acid or 51 monomers present into it. Proteins can be classified in two different forms based on their molecular shape, my dear students. Number one, it is known as the fibrous proteins, and number two, they are known as the globular proteins. Fibrous proteins, when polypeptide chains run parallel and are held together, by the hydrogen and disulfide bond, then fiber-like structure of protein are formed. So please remember, my dear students, that about the fibrous proteins, if we really go, when polypeptide chains run parallel and are held together by the hydrogen bond and disulfide bond, the fiber-like structure of protein is formed, and such proteins are known as the fibrous proteins. Once again, when polypeptide chains run parallel and are held together by the hydrogen bond and disulfide bonds, the fiber-like structure is formed, and such protein is known as the fibrous proteins. The examples of fibrous proteins are the keratin proteins and myosin. So keratin protein and myosin are the example of fibrous proteins. Please remember that these fibrous proteins are insoluble in water. Keratin protein is the example of fibrous protein which is found into our hair. It is also found into the wool. It is also found into the cells. And myosin is the protein that is found into the muscles of the living beings. Now we want to study about the globular proteins. When chain of polypeptides are coiled around to give a spherical shape, globular proteins are formed. Once again, when chain of polypeptides are coiled around to give spherical shape, the globular proteins are formed. So when the chain of polypeptide, they are not parallel, but they are coiled and they give the globular shape, such proteins are known as the globular proteins. Globular proteins are water soluble. Please remember that these globular proteins are soluble in water, and their globular proteins are water soluble proteins, and the examples of globular proteins are insulin and albumin. Please remember, my dear students, the examples of globular proteins are insulin and albumin. Structure and shape of the proteins are studied at 4 different levels. Now, please remember that insulin is the protein that is present into our body, which is utilized to digest the fats, to digest the glucose or sugar in our body, and albumin is present into the egg. You might have studied even about the blood, that the blood has got serum albumin. So this albumin is present into our blood also. This albumin is present also into the egg, my dear students. Now we want to study the various structure and shape of the proteins. The proteins have primary structures, secondary structures, and tertiary structures, and quaternary structures. So let us study about the various structures of proteins, each level being more complex than the previous ones. First, let us study about the primary proteins. Primary structure of proteins, it is the sequence of the amino acids in the protein structure is called the primary structure of proteins. So structures of protein sequence of amino acid in the protein is known as the primary structure of the protein. Each protein has a definite sequence of amino acids. Any change in this structure will cause change in the nature of the proteins, or new protein is formed if we change the primary structure of the protein. Once again, if there is a change in the structure of the primary protein, the new protein is formed from the old protein, or the protein, or it will cause the change in the nature of the protein. So this is how, my dear students, if there is change into the primary structure of protein, the new protein is formed. Secondary structure of proteins, my dear students, now we want to understand. The secondary structures of protein refers to the shape in which long polypeptide chains are arranged, the way found to exist in 2 different shapes. So secondary structures of protein is found to have 2 different shapes. First shape is known as the alpha helical shape. Second shape is known as the beta plated sheet or beta plated structures. Alpha helical structure, my dear student, alpha helical structure is the structure in which polypeptide chains coiled in a right-handed screw shape, in which Each coiled portion has a hydrogen bond between the CO and NH bond. This you can see here in this figure, my dear students, that they have a coil, and in this coil there is a hydrogen bond between the NH group and OH group. You can see here COO and NH, they have this hydrogen bond. Let us understand this with a better figure. Let's pay attention to this figure, my dear students. In this figure, you can see that there is a coiling of the structure of protein. You can easily see here there is a coiling of the structures of the polypeptide chain, and you can see this hydrogen bonding formed between the CO and NH group present in each coil. And this is what it is known as the alpha helix structure. Please remember, this structure is known as the alpha helix of the protein, and this is the secondary type of protein that we can understand. Now let's move and understand the secondary protein once again, so that we can have a clear idea about the secondary protein. Now you will understand it better. Secondary protein, it represents the shape in which the long peptide chains are arranged. They found to exist in two different shapes. First one is known as the alpha helical. Let us understand the alpha helix structure. Alpha helix structure is the structure in which polypeptide chain coiled in a right-handed screw shape, in which each coil or each portion has a hydrogen bonding between CO and NH. Now, my dear students, we are going to study about the beta-plated sheet. The beta-plated structure in beta-plated structure, instead of the coiling, here in this structure, polypeptide chains are arranged in the same plate, forming the hydrogen bonds among the C double bond O and NH group of the different polypeptides. The chains in this structure is very similar to the structure of the loosely folded clothes, or we can call it loosely folded cloth, or we can call it as a drapery. Please pay attention, my dear students. This is what it represents, the beta plated sheet, and in the beta plated sheet, my dear students, we can easily study the structure. You can see that here we have this RCH are arranged in a very linear manner, and then they are bonded with the bond with the CO and NH. You can see this CO and NH bond is bonded among this RCH in all the way, and this is what it is known as the drapery, or it is called the beta plated sheet structure of the secondary protein. So this secondary protein is found in two shapes. One is known as the alpha helix, and second one is known as the beta plated sheet. Beta plated sheet is like drapery, and here you can see that the coiling takes place according to the right hand move. So if you put your thumb upside and move your fingers into the fingers of the right hand, I'm talking about this. If you move your finger of the right hand, the way your finger moves, the same kind of coiling takes place into the alpha helix, and that is why it is known as the right-handed helix, or right-handed structure. Once again, I would like to show you that in alpha helix, the proteins are coiled according to the right-handed screw rule. Please remember that here the coiling takes place according to this right-handed screw way. So please remember that here we get the structure into the right-handed screw manner. You can see here. So now, my dear students, you are very clear about the secondary structures of proteins. Now let us understand the tertiary structure of proteins. Tertiary structure, it is the structure of the proteins formed when secondary structure is folded in a different folds. Due to these folds, the secondary protein become either globular protein or become fibrous proteins. So my dear students, the secondary structures is folded in such a way that the secondary structure turns out to be fibrous proteins or globular proteins. The remain force, forces which stabilize the secondary and tertiary structures of protein are number one. This is even an important point. The secondary and tertiary structures, the polypeptide chains are holded by either hydrogen bonding or disulfide linkage or van der Waals forces or electrostatic force of attraction. Please learn these four name of the force of attraction, my dear students, because they are very useful. Or many a time this question has been asked into the board exam even, that what are the forces that combines or that hold the polypeptide chain in secondary and tertiary structures? The answer is four type of forces. One is hydrogen bonding, another is a disulfide linkage, third one is van der Waals forces, and fourth one is electrostatic force of attraction. Now at last we want to study about the quaternary structures of the proteins. Some of the proteins are composed of two or more polypeptide chains referred to as the subunits. The spatial arrangement of these substances with the respect to each other is called quaternary structures. Please remember, some of the proteins are composed of two or more polypeptide chains referred to the subunits. So these two or more polypeptide chains are referred as a subunit. So we can say that some of the proteins are composed of more than two or more subunits. The spatial arrangement or the arrangement of these subunits in the space, which is known as the spatial arrangement. The spatial arrangement of these subunits with respect to each other is called the quaternary structures of proteins. So primary structures is the sequence of amino acid. Secondary structures is the alpha helical structure or beta plated structure. Tertiary structures either fold to give the globular proteins or fiber protein shapes. And quaternary structure is the spatial arrangement of the different polypeptide chains, which are two or more than two with respect to each other. So this is how, my dear students, we understand the primary, secondary and tertiary and quaternary structures of proteins. Now we want to study about the denaturation of proteins. Proteins are found in biological system with unique three-dimensional structure, and the biological activity of this protein is known as the name proteins. Once again, proteins are formed in a biological system with unique three-dimensional structure. The biological, and they have unique biological activity. Please understand, proteins formed in the biological system have unique three-dimensional structure and they have unique biological activity. It is these such proteins are known as the native proteins. When a native protein form, a protein is subjected to change or due to the change in the temperature or change in the pH or change due to some other chemical changes, the helix get uncoiled and the helix get uncoiled, unfolded and unopened, and the protein loses its biological activity. This is called denaturation of proteins. Now let me explain it once again to you, my dear students, that generally proteins found in the nature, they have unique structure and they have unique biological activity. But if these proteins are subjected to change in temperature, change in pH or some other chemical change are allowed to these proteins loses their biological activity because their helix and their polypeptide chains are uncoiled, unfolded, and due to this, these proteins loses their biological activity, and this process is known as the denaturation of protein. During denaturation of protein, secondary and tertiary structures are disturbed, but the primary structure is not disturbed. Here you can see in figure the primary structure, the secondary structure, the tertiary structures and the quaternary structures of proteins, my dear students. And you can even see these proteins are available in these forms in our body. The primary, secondary and tertiary structures of quaternary structures of protein you can see here. And here you can see that in primary structures, the part which is of a blue color is your part of carbon, and with the blue color, the dark blue color is your, sky blue is carbon and dark blue is your hydrogen. The red part represents the nitrogen, and this part, which is black in color, represents your oxygen molecule present into the structures. In tertiary structures, the green part is R group. You can see these green parts, and the red part is all your heme group, and this is the quaternary structures of protein that you can easily understand, my dear students, with the different figures. Same way, my dear students, but when denaturation of protein takes place, the primary structure remains the same, but the secondary and tertiary structures are affected. They get uncoiled, they get unfolded, and this is how we understand the denaturation of protein, my dear students. The denaturation of protein, the examples are coagulation of egg while boiling. So when we boil the egg, the denaturation of protein takes place and the egg albumin get coagulated. Same way, my dear students, when our milk is converted to curd, there is denaturation of protein takes place. So this is how, my dear students, we understand the various concepts about proteins. Now, if you have any doubt, you can ask me, my dear students. Do you have any doubts?
No doubt, sir.
No,
Okay,
sir.
then,
No,
let's,
sir.
let's finish the
No,
class
sir.
discussion here, and let us start revising the various